Today we will be reviewing over how to solve simple Mendelian genetics problems. These will include incomplete dominance, monohybrid cross, dihybrid cross, sex-linked characteristics, and multiple alleles. Before we begin reviewing over how to solve the genetics problems, we need to go over 4 genetics terms. Now, understanding these genetics terms is going to be key to success in solving genetic problems. These terms include heterozygous. Now, heterozygous means that the alleles are different. For example, if a person had brown hair, they would be big H, little h if they were heterozygous, having one allele for brown hair and one allele for blonde hair. Homozygous means that the alleles are exactly the same. So a person who's brown-haired would have an allele for brown hair from one parent and an allele for brown hair from the other parent. Likewise, an individual that is blonde-haired would have the blonde-haired trait given by one parent and the blonde-haired trait given by the other parent. So heterozygous and homozygous will always be stated in the problem when we're looking at the parents, and it's going to be key to understanding how to identify this in order to set up our problem. Phenotype and genotype are the next 2 terms we need to review over. Phenotype is what the trait exhibits. For example, we look at an individual, we can tell their hair color, brown hair. That would be an example of phenotype. Where genotype is what genes are present in order to give that hair color. For example, homozygous brown-haired or big H, big H. So before we go on, when we look at these, homozygous means that the letters are exactly the same. Here we would have homozygous dominant, big A, big A. Here we have homozygous recessive, little a, little a. Here we have heterozygous dominant, big A, little a. So in order to have a trait that is dominant exhibited, we just need to have one large A. So we can be homozygous dominant or heterozygous dominant. In order for a recessive trait to be exhibited, we need both recessive traits present. In other words, the organism would need to be little letter, or little a, little a. When we look at genotype versus phenotype, phenotype is just what we see, purple flowers or white flowers, where genotype is the letters that gave us the purple flowers, big A, or big A, little a. For white flowers, it would be little a, little a. So review over these 4 terms, make sure that you're comfortable with these before you continue forward. Before we go on and look at our genetics problems, we need to review over the gametes. Now remember, gametes are sex cells. So in females, these are the eggs, and in males, these are the sperm. A normal human has 46 chromosomes, so their diploid number is 46. Therefore, their gametes would have 23 chromosomes. They would be haploid. They carry half the genetic material from the parent. So as we look at how we determine our gametes, there are 2 ways we can look at determining gametes. One will be a FOIL method, very similar to math, and the other is called a fork line method. Now let's look at how we would determine a FOIL method. With our FOIL method, we would take our first in and our first out, and so we would have A, big B as a possible gamete. Then we would go A over to little b, and there we have a gamete. And then we come over here, and we would have A, big B. And then we would come over here, and we would have A, little b. So those are the possible gametes that we would have for a little a, big B, little b. Now, when we get to something where we're dealing with 3 or more traits at a time, the fork line is much easier to do. And so let's look at how we would do a fork line method. Our choices we have are a big C or a big C. Then we have a big D little d, and both can give big D or little d. And then we look and then we have a little e or little e. And we'll notice that some of these are repeats. So we're going to ignore the repeats. That means that this right here will be ignored because it's a repeat of above. And that means this one right here will be ignored because it is also a repeat. And this right here would be ignored because it's a repeat. So our gametes would be C, D, E, and C, D, little e. And so those would be the approaches that we would have in order to solve these problems. Now I have given you 3 additional practice problems, and you'll find the solutions on the next page. So as you went through and you solved your gamete problems, hopefully you had 4 answers when you were looking at the number 3, the A, B, little b, big C, little c, D, D. For number 4, there would have been 32 possible answers. That one may have been a little difficult. And then number 5, you would have had 2 answers. So do practice over fork line, go over a FOIL method. FOIL method, again, is going to be best when we're dealing with 2 traits. And the traits are the letters. So we have A's and we have B's. Now when we have 3 or more traits, which are the C's, D's, and E's, that's going to be using our fork line. And just remember that if we have a repeat, we don't need to write that down. So on number 5, we have U. Well, we have this U, it's the same, so we're going to ignore it. We have V, and we have that V, it's the same, we're going to ignore it. We have W, and then we have that W, it's the same, we'll ignore it. X, and X is the same, we'll ignore it. Y, well, right there, we'll ignore it. And then we had Z, and we had little z. So we would have had UVWXY, big Z, UVWXY, little z, as our possible choices. Now as we so a quick way we can determine if we have written out all of the possible gametes is going to be to use this formula right here, 2n. Now n is the number of heterozygous. So if we look at number 1, we'll notice that little a is homozygous, big B, little b is heterozygous. So we would take our formula 2, and of these we only have 1 that would be heterozygous, so 2 raised to the first power, we would have 2 gametes, little a, big B, and little a, little b. On number 2, we look at this and we have just one set of heterozygous, the D's. So again, we would take that 2 raised to the first power, and we would have 2 possible gametes, C, big D, E, and C, little d, E. Now on 3, we look at this, and we'll notice that we have the B's and we have the C's. So this time, we will take 2 raised to the second power, and that would be 4 possible gametes. On number 4, we have the M's, the N's, the O's, the P's, and the Q's. So 2 raised to the 5th power, we would have 32 possible different gametes. On 5, when we look at this, we only have the Z's as our heterozygous, so again 2 raised to the 1st, and we would have 2 possible gametes that we had listed below. So use the 2 to the nth power to determine your gametes and to ensure that you've written out all the gametes before you continue your problems.